Hi, my name is Tiffany Velasquez, and this is my Unit 2 Case Connections assignment. And for this assignment, I chose to do case number 2 and case number 4. So I will start out with case number 2 first. We'll jump right into it. So the first question that we come across is how glucose uptake is taken into normal tissues. So glucose uptake is regulated in the liver by GLUT2, into the skeletal muscle by GLUT4, into the brain by GLUT1 and 3, and into adipose tissue by GLUT4. The next question we come across is how glucose uptake into normal tissues compares to the glucose uptake into tumor cells, and also why this is an advantage for these tumor cells. So cancer cells are super selfish and super greedy. So they alter energy metabolism by increasing glucose uptake. And they are essentially stealing glucose from other cells. And this is an advantage to them so they can grow and thrive and spread and further metastasize. This glucose uptake is seen in the image provided on the Unit 2 assignment. You can see that in the tumor cells when the patient was given glucose, that you can see that on the imaging of the patient. So through doing research on the subject, I came across the Warburg effect, which is worth mentioning. It's essentially just talking about the rewiring of metabolism that happens, and the increase of glucose uptake, and it explains the increase of glucose uptake and the fermentation of glucose to lactate and how that works in these tumor cells. So the modifications that, the next question is what modifications may have occurred in the cancer cells to alter this glucose uptake that we see. And the modification that allows these cells to increase the glucose uptake resides in the fact that there are an increased amount of GLUT receptors on these tumor cells. And that's why there's an increase of glucose there. I did also read that the changes of the glucose metabolism in tumor cells are also enabled by what are called oncogenes. And these genes stimulate the uptake of glucose, alter oxygen consumption, and limit the response to oxidative stress and DNA damage. So essentially, these genes are making these tumor cells even more resilient than they already are, as mentioned earlier, to grow, spread, metastasize, and do what tumor cells do. So that concludes my case number 2. So I'll move on to case number 4 now. And from what I have gathered from the scenario of our patient is that The administration of epinephrine should have raised the glucose levels, but since the lactate levels rose instead, this shows that there is a problem tapping into the glycogen storage. So I concluded that the answer was that the patient has glycogen storage disease, which alters the way that glycogen is used and stored in the body. It's due to a missing enzyme that is typically used to break down glycogen, and this leads to a buildup of glycogen in the liver. And this is consistent with the patient's symptom, such as enlarged liver, hypoglycemia, and the inability of the patient to maintain glucose concentration while fasting. The next option is glucose 6-phosphate dehydrogenase deficiency, and from now on I'll just say G6PD deficiency, which is a genetic condition that mostly affects African American males, which is not consistent with our patient. It causes red blood cells to break apart prematurely, resulting in hemolytic anemia. Signs and symptoms include jaundice, enlarged spleen, sleepiness, and these symptoms tend to rise when exposed to a trigger. And some triggers include certain foods or medications. It has been found that people with G6PD deficiency have impaired fasting glucose, and they're at an increased risk of diabetes. However, this finding is inconsistent with our patient, as G6PD deficiency would result in hyperglycemia, which is consistent with diabetes, with that finding. G6PD is the first step. It is the first step in the pentose phosphate pathway. And without this enzyme, levels of NADH would decrease, and the oxidation of glucose cannot occur. A simple blood test could rule out this condition, and it's just worth mentioning that G6PD, the gene for G6PD, is found on the X chromosome. As mentioned earlier, mostly males are affected. So next we have galactosemia, and it is a metabolic condition that results in the inability to metabolize galactose, which is often found in milk products. This leads to a buildup of galactose in tissues and in the blood. It can result in liver failure and mental deterioration. Characteristics of this condition are inconsistent with hypoglycemia and symptoms found with our patient and can be ruled out. And the last one is hereditary fructose intolerance, which is a result of the absence of the enzyme catalase B, which cleaves fructose 1-phosphate. Symptoms include hypoglycemia and vomiting after consuming foods high in fructose, And this condition can be ruled out because breast milk contains galactose and not fructose and wouldn't be affecting our patient in this way. And the mother has stated that she has not given the child anything other than breast milk since birth. Yeah, so that's my assignment. Thank you for watching.